Hello, this is Ankita, and today we are going to study about the prokaryotic cell. Prokaryotic cell is the primitive cell, right? It was formed in the beginning of the life, and from prokaryotic other cells arrived. Prokaryotes are the microscopic unicellular microorganisms which has neither a distinct nucleus with a membrane nor a specialized organelles, right? So prokaryotes are the microscopic, that they can't be viewed without microscope. They are unicellular, that is they are made up of one cell only, they are never multicellular, which has neither a distinct nucleus, that they do not have a proper developed nucleus, because they have nucleoid instead of nucleus, and that nucleoid is simply compact genome of the bacterial cell. It is not enclosed in any nuclear membrane, and it also do not have any other specialized organelle as in case of eukaryotes, right? As eukaryotes have various specialized organelles, so in case of prokaryotes they do not have any specialized organelles within them, right? And the examples of prokaryote are bacteria and cyanobacteria. Bacteria, as you know it very well, and cyanobacteria is the blue green algae. So let's have another slide. Yeah, there are some features of the prokaryotes. The examples of prokaryotes are bacteria, mycoplasma, PPLO, and blue-green algae. Mycoplasma is the wall-less bacteria, you can call it, right? So the mycoplasma is also very harmful. It also causes various diseases, and the PPLO is pleuropneumonia-like organism. It is also a type of prokaryote. Blue green algae, that is cyanobacteria, right? So bacteria, mycoplasma, PPLO, and blue green algae are the some examples of prokaryotes. They are smaller and multiply more rapidly than the eukaryotes, right? So as we know it very well, of course prokaryotes are very smaller in size as compared to eukaryotes. I have told you in my earlier video, in where I have explained you differences between eukaryotes and prokaryotes. I have told you that the size of prokaryotes ranges from 1 to 10 micron only, while the size of eukaryotes ranges from 10 to 100 micron. So you can see the difference in size, and of course they multiply more rapidly than the eukaryotes because of their smaller size, and they do not have various components for their replication. They do not have very much work as compared to eukaryotes, right? Cell shape are bacillus, coccus, vibrio, and spirillum. Bacillus, that is they are rod shaped. Coccus, that is they are spherical. Vibrio, that is they are comma shaped, and spirillum is something like this, right? So these are the some shapes of bacterial cells. Yeah. You know the examples of bacillus. You can example of bacillus is lactobacillus, coccus is lactococcus, and vibrio is cholera vibrio, right? So these are the some examples of bacteria also on the basis of their shape. They have cell membrane which is made up of phospholipids. They have phospholipid as their cell membrane. Phospholipid bilayered structure, right? Do you know how is it formed? Phospholipids are the structures having this phosphate group and this fatty acid chain. This phosphate group is hydrophilic. It is hydrophilic, while these fatty chains, they are hydrophobic. They are okay, hydrophobic. Yeah, they are hydrophobic. So they repel water in their contact. Whenever they come in contact with water, they repel, right? As you know, lipid, for example, oil, that also repels water. That is not towards water. If you put a drop of oil on the water, it will float. It is not going to mix with that, right? In the same way, the phospholipid structure, where lipid, that is a fatty acid that repels water, that is hydrophobic in nature, while the phosphate, that is hydrophilic in nature. And they have fluid filled inside the cell membrane, that is called as cytoplasm, right? So the fluid filled inside the cell, that is cytoplasm. And the composition of cytoplasm in case of eukaryotes or in case of prokaryotes, it is most probably similar, right? It is a different thing that some enzymes will differ, but they have a bit the basic composition is same in case of either eukaryotes or in case of prokaryotes. They do not have a well defined nucleus because it lacks nuclear membrane, and rather it have a nucleoid as the compact genome in the cytoplasm. So if we see yet that yes, this is the bacterial cell, and it do not have a proper nuclear membrane. It has genome like this only, right? It has nucleoid form. It has genome in the form of nucleoid. It do not have any proper nuclear membrane to enclose it, right? So that's why it is a simply membrane. You call it. The cell without nucleus, right? It do not have a proper well developed nuclear membrane, that's why it is not having proper nucleus. Yeah, and they have plasmid as the extra chromosomal genome present in the cytoplasm. Plasmid provides many extra features as like resistance. Yes, of course it provides resistance, and it also acts as a vector in the genetic engineering. Yeah, it acts as a vector. It provides resistance and it acts as a vector in the genetic engineering, right? These are the 2 points that I need to mention. That plasmids are, of course, they are not part of the main genome of the bacterial cell, because the main genome that is responsible for the life of the bacterial cell, if they're not going to have their main genome, they won't survive. But the plasmid that is not going to make them make, that is not compulsion for the survival of the bacterial cell, but that provides some extra features to the bacterial cell, for example resistance and various other features in competition also with their surrounding bacteria, right? Resistance in case of antibiotics that we take. So if we keep on taking the same antibiotic, so the bacteria will get resistance towards against that particular antibiotic, and the next time after many times use, the next time when you will use it the same antibiotic again, so bacteria will be very much resistant against those antibiotic, and it won't get killed by that antibiotic, right? And you won't get any relief. So they have plasmid for the resistance, right? So the resistance comes from the plasmid. And it also acts as vector in the genetic engineering, right? It also acts as a vector in genetic engineering. So vector, as we know it very well, that vector is used for transfer of required fragment from one source to another, right? Especially from media to the bacterial cell. So it also acts as vector in the genetic engineering. And no organelles in eukaryotes except ribosomes, which is 70S type. They have ribosomes which is of 70S type. So you know what kind of ribosome they have? They have ribosome like this is the smaller unit and this is the larger unit, right? So the smaller unit that is of 30S type, while the larger unit is of 50S type. The S resembles Sevedberg unit. It resembles Sevedberg unit, and that signifies the coefficient where Sevedberg coefficient, and this Sevedberg coefficient denotes that at what centrifugation will these ribosomes get precipitated, at what centrifugation you will able to remove out these ribosomes from a cell suspension, right? So simply for you it is enough to understand that yes, this whole ribosome, this 730S and 50S, these subunits together are making 70S type of ribosome in case of prokaryotes, right? And in case of eukaryotes, when we will study about eukaryotes, I will explain you over there. So it has a smaller unit as the 30S and a larger unit as the 50S, right? Wait, yeah. And mesosome in the cell membrane folding, which is found as a vesicle, tubule or lamellae in shape. So they also have mesosomes in the cell membrane, right? In the cell membrane, that is the bacteria also have mesosomes, which are simply the cell membrane enfoldings, and those enfoldings form a shape of vesicle, tubule or lamellae in shape, right? So vesicle, of course, they will have a vesicle like this circular vesicle, lamellae like a finger, I mean like this, lamellae or tubular, that is of course like this. So in this shape you can get mesosomes in the bacterial cell, right? Sorry, yeah. So it is about this page, yeah. This is the diagram of prokaryotic cell. Here it is shown capsule, ribosomes, the cell wall, plasma membrane, the nucleoid, flagellum, inclusion body, chromosome, DNA, and this fimbriae. It is a simple diagram of bacterial cell, right? They have a nucleoid in the center, and they have plasma membrane. Over the plasma membrane and they have cell wall, right? Over the plasma membrane they have cell wall also. Let me have a proper color. Yeah, can I show you? Yeah, this is the plasma membrane. This brown one is the plasma membrane, while you can see that this light yellow one is the cell wall, and above this we have very well outer membrane, or you can say that is the capsule, right? This outer one, that is acting as capsule, that is the capsule. I'm not able to erase it. Yeah, so this is the capsule or outer one. So these three things are going to create the bacterial envelope, which provides protection to the bacterial cell, right? So this is the simple diagram of the bacterial cell. Ribosomes are present in the cytoplasm. They are floating in the cytoplasm. They do not have any organelle to get attached. It is about the cell envelope, as I have shown you earlier. That cell envelope act as protective unit. It act as protective unit in for the bacterial cell, and this single protective unit is made up of glycocalyx, cell wall, and plasma membrane. It is made up of glycocalyx, cell wall, and plasma membrane. Glycocalyx differ from bacteria to bacteria, and it is of two types. It could be slimy layer, in the form of slimy layer, or it could be in the form of capsule. So if it is slimy layer, so it is loose sheath of glycocalyx, right? So when the glycocalyx, the outermost layer of bacterial cell, if it is present in a loose form, so it will be a slime layer. But when it is thick tough form, it is the glycocalyx, right? It is the glycocalyx when it is in tough form. But the moment when it is in loose form, it is only slime layer. The inner one, the second one, is the cell wall. Cell wall is made up of peptidoglycan. It is made up of peptidoglycan. It determines the shape of the cell and provides a strong structural support to prevent the bacteria from bursting or collapsing, right? It prevents the bacterial cell from bursting or collapsing. What this bacterial this cell wall, right? Cell wall is made up of peptidoglycan. Cell wall is made up of peptidoglycan. It determines the shape of the cell, and it provides a strong structure support to prevent the bacteria from bursting and collapsing. Bursting means it do not allow it to burst when the concentration of the solution in its surrounding is differ from than it required normally, right? And thickness of the cell wall thickness of the cell wall and reaction towards the gram stain confirms gram positive and gram negative bacterial cell, right? So the cells which are gram positive, they have thicker peptidoglycan, while the cells which are gram negative, they have thinner peptidoglycan, right? So on the basis of the gram staining and on the basis of the thickness of this bacterial cell wall, they get divided into gram positive bacteria and gram negative bacteria. And plasma membrane, it is phospholipid bilayer structure. It is semi-permeable in nature, right? Phospholipid bilayer structure, that is, the structure is something like this. No, let me have a pen. Yeah, yeah, 1 2 3, 1 2 3. These are the lipids, and yeah. This is the phosphate, the head, and this is the tail portion. This is the tail portion, which is, yeah, this is the tail portion, which is fatty acid, or you can call it lipid, right? Yeah, so this is phospholipid. This is phospholipid. This is That's why it is called as bilayer, because one phospholipid molecule and this is another phospholipid molecule. So when the plasma membrane is formed, they are like this, overlapping each other, right? So two layers of phospholipid are there. That's why it is called as phospholipid bilayer structure. It is called as phospholipid bilayer structure, right? Yeah, and it is semi-permeable in nature because it cannot allow every molecule to enter inside the cell at the same time when it is not requiring those components, right? I have explained you in my earlier videos also that plasma membrane is semi-permeable, or you can call it selectively permeable, that it do not allow every component to enter at any time. It allows a particular component only when it requires, right? This is the mesosome structure. Yeah, here is the mesosome structure. The unfolding of the plasma membrane. It is the plasma membrane unfolding, vesicle, tubular or lamellar in shape. I have explained you. It helps bacterial cell in cell wall formation, DNA replication, secretion process, and also increase the surface area of plasma membrane for various enzyme activity, right? Of course, if it get folded, so it is going to give more and more plasma membrane area for more functioning, right? So it helps in bacterial cell wall formation, it helps in DNA replication, it helps in secretion process, and also increase the surface area of plasma membrane, for that helps in increase of various activities of bacterial cell, right? But in case of blue green algae, it has chromatophores, which are membrane extensions in the cytoplasm. So instead of mesosomes, they have simply chromatophores. They have simply chromatophores, which are the membrane extensions in the cytoplasm, and those chromatophores are responsible for their photosynthesis, right? That help them in their photosynthesis. Locomotion, that is whether how they locomote or how they move. So some bacteria have locomotory, whilst some are not, right? So some bacteria are locomotory because they have flagella for their locomotion, while some are not locomotory because they do not have flagella for their locomotion, right? Flagella is consist of filament, hook and basal body, where the filament is the largest part, and fimbriae and pili are some other examples. So fimbriae are the small bristles for attachment to the host surface, while the pili, which is made up of pilin protein, uh, for example, sex pili is an example which plays a role in conjugation. So pili have different roles for, but this fimbriae, it has a special role in a small. They have a special role in attachment to the host. The moment till the bacteria is not going to attach to a surface or the host surface, it is not going to infect the host, right? So attachment is must. Ribosomes, they are, I have explained you that 70s type of ribosome. Yeah, 70s type of ribosome, 50s and 30s as subunits, and size is the 15 by 20 nanometer, right? So they have 15 by 20 nanometer in size, and they are very small in size. It is only site for translation, that is protein synthesis. Protein synthesis can occur at ribosome only, and polyribosomes are the structures which are formed when many ribosomes get attached with a single mRNA. So in that case, the translation will occur at very higher speed, and that is also known as polysome. Yeah, and there are some inclusion bodies. Inclusion bodies are some vesicle-like structures which are used for storage purpose. For example, this phosphate granules, cyanophagine granules, and glycogen granules, gas vacuoles structure, these all provide some extra features to the bacterial cell for their survival. They help in their survival in some drastic conditions. Carboxysomes just to store some enzymes which help in their photosynthesis. Phosphate granules, and these are the different type of granules which are required, which are present in different type of bacterial cells on the basis of their requirement. Yeah, and here is the picture that I have given you to get some extra points, so you can take a type of brief summary from this. picture, right? Thank you so much for watching this video. If you have any queries, you can ask me in the comment section. Thank you.